Okay, ma'am.
Okay, good afternoon, all of you, beta. Now, today's session is your seminar session, and now over to Ritika. Ritika, let's start.
Good morning to all of you present in this class. I am Ritika Singh and I am from class 9th A. This class is merged with class 9th A and with class 9th B. Today is our seminar presentation and we are going to explain the chapter cell which will be conducted by each student which had raised the hand. I will also explain a chapter or a topic. So, ma'am, I will ask you to please give permission to start.
Yes, beta. All of you start your session.
Oh, as an anchor, I will ask the raised hand student who will start the first. So, if anybody want to start this seminar firstly, so they can. Please let me know. Is anybody is here? Okay, nobody. So, ma'am, let me start.
Yeah, beta.
Okay. Um, Thank you, ma'am. Okay, ma'am.
Hmm.
As I have given my introduction that I am discussing, and today I am going to explain chapter or topic endoplasmic reticulum, which is a organelle which is present in cytoplasm. So let me firstly share the screen. So yes, so this is the endoplasmic reticulum. What is the meaning of actually the endoplasmic reticulum is? So endo means inside, plasmic means cytoplasm, and reticulum means a network. So we can say a network inside the cytoplasm. The definition of ER is, as we can say endoplasmic reticulum as ER in short form. So ER network of flattened sacs and tubules in cytoplasm. So the cytoplasm is divided into two parts. The first one is this one is the rough endoplasmic reticulum, and this one is the smooth endoplasmic reticulum. This portion is not, for your kind information, this is not a endoplasmic reticulum part, it is a nucleus. So the where the actually endoplasmic reticulum start. So there is a, you can see here a short space. So it is a part of nuclear envelope. So there is a perinuclear space from where the actually endoplasmic reticulum start. And further, you can say here you can see a stalk sacs which is known as cisternae. On this cisternae, this sac like a structure, there is ribosomes present, which is also a type of uh, mm, or you can say organelle, and it is a factory of protein. Because of ribosome occurrence, it is known as rough endoplasmic reticulum, and there is a smooth endoplasmic reticulum. Here ribosomes are not present, so they are called smooth endoplasmic reticulum. Here you can I have said that the ribosome is a factory of protein, so it releases protein, and the smooth endoplasmic reticulum produces lipids and many more enzymes. So the there are three occurrences in endoplasmic reticulum. These are the first cisternae, the second is vesicles, and third is tubulus. Okay, the what is cisternae? I have told you that the this sac-like structure is known as cisternae. They are long, unbranched units and arranged in stalks, as I have told you all. So its diameter is about 40 to 50 micrometer. And yes, the endoplasmic reticulum thickness is about 5 nanometer. Now, the second is vesicles and vascular form. The vesicles or vascular form is very important because actually they are a oval membrane or bound structure which are in oval shape, which they are the like this. It has structure like this, and they are the synthesized product molecules which are synthesized by the endoplasmic reticulum, like the protein, which is synthesis synthesis molecule which is conducted by rough endoplasmic reticulum. Now, so and you know where the endoplasmic reticulum and start and where it end. So the endoplasmic reticulum starts with this means with the nuclear envelope, and it is attached with cell membrane. And as we know, the present one is the cytoplasm. It is in cytoplasm. Okay, so this was all. Now, there when the vesicles are produced or offspring, so on when the cisternae have to offspring, or sorry, off disattach the vesicles, there is bud form, and they disattach the vesicles. So now we will get about knowledge about rough endoplasmic reticulum, or it is also known as granular ER, because ribosomes are present here. Ribosomes attach to its membrane. Here the ribosomes are present, and it is attached with ribofuscin protein, a protein named with ribofuscin protein. It helped to attach the ribosomes with the cisternae. The second one is the RER attached to nuclear envelope forms direct connection between perinuclear space and the lumen. What is lumen? So the space between the cisternae is known as cisternae space or lumen. So the movement of molecules is there, as we have know that nucleus is porous to exchange the gases and the necessary components which is wanted by them. They get transported with each other. Now, as ribosomes are present in rough endoplasmic reticulum, it is important in synthesizing, packaging, and secretion of protein, and abundantly found in liver cell and pancreas cell, etc. So the second, as I have told you, that the smooth endoplasmic reticulum, RER, it is known as a granular ER because ribosomes are absent. So it is composed of tubules involved in metabolism of lipids, including steroids and glycogen for their storage and their storage too, and abundantly found in adipose tissues, glycogen storing cells of liver and spermatocytes, etc. So this was about the endoplasmic reticulum. What are the functions of endoplasmic reticulum? So the ER act as intracellular circulatory or transporting system. As we have seen that there are exchanges of many things here, and this is a very large process, so we can say it intracellular. Cellular means it is present in cells, so intracellular circulatory, and a transporting system, as many substances is transported from one side to another. RER pinches of certain teeny proteins, which is known as vesicles, which is filled in vesicles, which ultimately fuse off Golgi bodies. Then the vesicles are go to the Golgi apparatus. The ER is a part of endomembrane system or cytoplasmic vacuolar system. The mechanical support, this mechanical support gives the shape to the cell, and this endoplasmic reticulum have many enzymes, and it is known as also a site of biogenesis of cellular membrane. Biogenesis means formation, so it helps in the cellular membrane, means cells membrane. And for new nuclear membrane envelope after each nuclear division. When the nuclear division, you will get it next. When the nuclear division is done, the nuclear envelope, the cistern, this all also divided and makes more amount of endoplasmic reticulum. And the most important function of endoplasmic reticulum is in drug detoxification or poisonous. Detoxification means the removal of toxic substance which is not good for our health, and it is done by the endoplasmic reticulum. And there is a sarcoplasmic reticulum. It is also like a it is another form of, you can say, it is look like endoplasmic reticulum. So it is mainly in cardiac, it is present mainly in cardiac cells or muscles cell, where it is used more, and it stores ions. This sarcoplasmic reticulum stores ions and calcium, ions means like most of the calcium ions. So this was all about the endoplasmic reticulum. And one more thing, that nucleus, nucleus have the mRNA. So mRNA, a messenger RNA, which is go through this endoplasmic reticulum and give the messages to do that all work. This is very important organelle and a very big organelle. So this was all about endoplasmic reticulum. Hope you all understand it very yes. So
Very
let
good,
me show
Ritika.
you.
Very good, Ritika.
Thank
Very
you, ma'am.
well presentation. And your explanation is also very good.
Thank you, ma'am.
Okay, Beta.
So as a conductor of this seminar, I would ask to, ma'am, may I choose the student who will
Yeah,
like?
Beta.
Give?
You choose
Oh,
the
okay, ma'am.
students.
Okay, ma'am. Naman Sharma, will you give this presentation next?
Yes, I
Okay.
will.
So I am handovering this to you.
Yes, ma'am. My name is Naman Sharma. I am from 9th B, and I am here to tell you everyone about the cell and explain. I am going to explain it. Now we are know the cell. It is the basic structural, functional, and biological unit of non-living organism. Only non-living organism, मतलब जिनके बारे में तो हमें पता ही नहीं, तो हम कैसे बता सकते हैं उनके पास सेल होगी. तो a basic structure. Now basic structure, what mean the basic structure? There we have study about the cellular level organization. In this, the cell is the smallest unit, then the cell combines and make tissues, and then tissues combine and make organ, and organ combine and make organ system, and then organism or any living organism formed. And what is the functional? Every necessary functions which are required to perform all the basic needs are found in these cells. many persons are confused that it is the smallest part. It is not the smallest part. It is the smallest functional unit, basic unit. What is the difference between smallest part and unit? The part means वैसे तो very smallest part is ribosome in the cell. It is also smaller than that this, but the smallest unit means it can perform all the tasks. The scientists have divided called it a smallest unit because this can perform all the important tasks which are needed in our body. The scientific name of cell is cyto, uh, and the study of cell is called cytology. The cell was discovered by the Robert Hooke in 1665. It was the cork cell which was dead, and the living cell was discovered by Antony von Leeuwenhoek. Uh, he discovered the bacteria or living cell flowing into the water with his made microscope. Now we are going to study parts of cell. There are many parts of cell like cell membrane. It is the outer covering of any cell. Any cell we can see the cell membrane. Next, cell wall. Cell wall is found in plant cell and some chosen bacteria. And next is protoplasm. Protoplasm has two parts, cytoplasm and nucleoplasm. Nucleoplasm present in the nucleus, and cytoplasm present in the outer of nucleus. Next is nucleus. It consists of nucleolus, nuclear membrane, and I mentioned here chromosomes. In this chromatin material is present. When it is time to cell division, then chromatin material convert into chromosomes, and this in this chromosome DNA is present, and in DNA there is gene present. And then cell organelles, the like Ritika has explained, the endoplasmic reticulum. Next, Golgi bodies. In endoplasmic reticulum, ribosomes are present. These are protein factory. Mitochondria, it is the powerhouse of cell. Lysosome, it is also called the suicidal bag because it clear the dirtiness of the cell. In this, some enzymes are present which can मैम जैसे मैम गला सकता है मैम कोई भी गंदी चीजों को जो सेल के अंदर मैम डेड होती हैं.
Which can melt.
Yes, and that next is chloroplast and food vacuole. Ma'am, in food vacuole there is 65% of water. It contains all the food of the cell which are needed. Next is types of cell. There are two types of cell: prokaryotic cell and eukaryotic cell. Prokaryotic cell, ma'am, in which there are not any well developed organelles, and in eukaryotic cell there are double membraned and very developed organelles. And next difference is that prokaryotic cell have nucleolus only and have DNA and some other materials, and in eukaryotic cell all the nucleus, these all the parts are present. Next, I am going to tell about the smallest cell. The smallest cell in the world is mycoplasma. The largest cell in the world. I am talking about in the world, not in the human body. I will tell it again what is the smallest cell and the largest cell in human body. The smallest cell is mycoplasma. Largest cell is called ostrich cell. The smallest cell in human body is the sperm, and the largest cell in human body is ovum, and the longest cell in the human body is neurons which are present in our brain. और मैम यहाँ पे lowest division it is given that lowest division, but I want to tell there is no division found in the neuron cell. That is why if we are injured in our brain, we cannot recover again. There are 90% of neurons and 10% of other cells. I don't know their name. And the fastest division is found in the liver cell. That's why our liver is only damaged, means they stop work. They're not very seriously damaged. That's all about what I have to tell.
Very good, Naman. Very good. Next,
Thank
Ritika.
you. Yes, ma'am. Thank you, Naman, for explaining us about cell. Thank you, everyone, to participate in the seminar, and for you all who had listened very carefully. Thank you, ma'am, for giving this chance to represent our information. And today's seminar was very good and very effective for all. Thank you so much to you all to came in this seminar. Thank you so much.
Very good, Ritika, and all of you. Now we end the meeting.